Hi everyone, my name is Talia Griffin, and I am an alumna of Albany State University, now entering as a first-year microbiology PhD student at Cornell University. As a participant in the International Research Experiences for Students, or IRES, program in Japan, I was assigned to the National Institute for Basic Biology in Okazaki, Japan, under the mentorship of Dr. Kiyoshi Naruse. Here, I was able to investigate genes within a gene regulatory network that was involved with sexual development in Medaka. I was able to utilize CRISPR-Cas9 and target our genes of interest in order to establish knockout lines. Additionally, we constructed plasmids with a novel fluorescent reporter. This reporter was inserted into Medaka embryos via CRISPR-Cas9 mediated knock-in. We were able to achieve this through a variety of techniques, some of which were new to me. These include microinjections with the Cas9 to enable genome editing, and heteroduplex mobility assays to measure the cutting efficiency of our Cas9 and detect the presence of mutations. We also conducted classic laboratory techniques such as PCR, DNA transformation, and Sanger sequencing. As someone who has received their training primarily within microbiology labs, this work was incredibly unfamiliar. However, by being invested in the literature and asking questions, I was able to piece the puzzle together and get excited about the project that we were working on. And that is all thanks to the enthusiasm of my mentor. While the research was critical and fast-paced, I was also able to foster connections between my mentor as well as the other lab members at NIBB. They were extremely caring, and they helped to facilitate my growth scientifically, as well as help me be more receptive to the culture that was around me. The Naruse lab was extremely patient with my less than stellar Japanese, and they were even able to help me develop stronger language skills over time. Being in such a tight-knit research group helped me to be more culturally conscious and adaptable to my surroundings. I was able to have experience with CRISPR-Cas9, which was what I was hoping for, and learn new techniques and analytical skills that are unique to the labs that are collaborating with this program. Overall, the Naruse lab felt like a second family. I am so grateful to the IRES program for placing me in such a warm and welcoming lab that allowed me to grow and strengthen my knowledge within genetics. Outside of lab, we were able to travel to so many different places and be immersed in the culture and scenery of Japan. I traveled to Kyoto, Osaka, Kanazawa, Hiroshima, Nikko, and various other locations across Japan. This allowed me to be surrounded by the history found within castle walls, the etiquette of shrines, and be able to participate in local festivals such as the Hydrangea Festival and customs such as kimono wearing or glass blowing. Experiencing Japan like this is naturally breathtaking with its mountains, hiking trails, and unique flora and fauna. This program allowed me to see beyond my own cultural perspective and worldviews, especially in regards to human interactions and social responsibility. The IRES program in Japan was a phenomenal experience, and it was irreplaceable in my development as a global citizen. I am so incredibly grateful to NSF, the program coordinators, and my Japanese host lab. Thank you once again for such a transformative experience.